That's it, right? So yeah, just so you know, this is a recorded meeting. And to start, I would like to thank the IBH, the coordinating committee of the IBH, for this very kind opportunity to discuss with you recent insights from our research on microbe interactions using barley as a model. So I'm Davide Bulgarelli from the University of Dundee, where I'm a principal investigator. And together with me today, there is Carmen Escudero Martinez. She's a postdoctoral scientist in the lab, and basically we will do this presentation together. Now, so what we are going to do today with you is providing you with an introduction of the plant microbiota, and in particular the microbiota that we like to, let's say, study, or we are interested in the most, which is the one at the root-soil interface. And then I will explain you why we think this is a strategic area of research from both basic science and translational application standpoints. Then I will illustrate some of the methodologies, the most common methodologies that can be used to basically investigate the microbiota. After that, I will provide you with snapshots of some of the recent investigation we have conducted using barley as an experimental model to study plant microbiota interaction in a sort of light touch. And I will cover more the conceptual basis of these experiments to the benefit of the audience that perhaps is less familiar with microbiota science. Towards the end of this second part, I will leave the stage to Carmen, who will delve into one of the most recent investigations we have conducted, bringing together basically microbiota science and plant genetics, and then she'll be in charge also of wrapping up and illustrate the direction the lab would like to take in the years to come. So if you have a question, there is a chat. I believe that you have a button on the bottom, on the right bottom side of the screen where you can type your question, or you can raise your hand at the question and answer session at the end of the presentation, and Carmen and I will try to address them the best. Okay, so let's start then. My lab is interested in global food security, and we believe that global food security passes at least in part to soil and microbes that in soil interact with plants. And in particular, we are fascinated by the thin layer of soil surrounding plant roots, which is designated rhizosphere. The rhizosphere, from chemical, physical, and biological standpoint, is markedly distinct from what is called bulk soil, so a piece of soil that is without plants. And within the rhizosphere, so the rhizosphere represents a microhabitat for a very distinct microbial communities interacting with plants that is defined as the plant microbiota, or the microbiota populating the root soil interface. At the same time, the rhizosphere represents a site for resource exchange. So plants are pushing at the interface between root and soil, in soil, organic compounds that have been fixed by a photosynthesis in a process that is called rhizodeposition. And at least in part, or I would say for a significant part, this rhizodeposition is responsible for the modification that we can see in the rhizosphere compared to unplanted soil. And in the rhizosphere, mineral nutrients become available for plant uptake, and therefore basically plays a critical role in plant nutrition. So, and by the look of it, I think that it's not difficult to imagine a parallelism between the situation occurring at the root soil interface and going on in our own digestive tract. So we have an area for, let's say, resource acquisition and microbial communities, a microbiota that can contribute or interfere with the activities of a multicellular organism. In analogy to the probiotic members of the gut microbiota, plants have their own probiotics that have been often referred as plant growth promoting rhizobacteria, or more generally, plant growth promoting microorganisms. Those microorganisms effectively capitalize on what plants can provide them in form, for instance, of the organic compounds that I mentioned before, and in return, they can increase access to mineral nutrients, they can protect can protect plants from potential pathogens or pests, in particular the one that can be proliferating in soil, and attenuate abiotic stress, attenuate abiotic stresses. So in a way, these members of the microbiota can effectively replace or at least let's say perform some of the functions that nowadays farmers often delegate to non-renewable inputs in the agroecosystem. However, the interaction between plant and microbes in soil can go terribly wrong. There are a group of microorganisms in the microbiota that is particularly greedy. They take from plants and give back nothing. So those are the causal agent of so-called soil-borne disease. And from ecological standpoint, they can be considered as a causative agent of dysbiosis of the microbiota. So the diversity of the microbes that is present in a healthy plant is gone because their microbiota becomes dominated by these greedy elements. So, and already from this very first introduction, I think it became clear that define the rule of engagement at the root soil interface between plant and microbes is absolutely crucial to ensure plant growth, development, and health, and ultimately sustainable crop production. Now, if you look at those rules of engagement, what we can infer from studies conducted by our group or many other groups is, despite the staggering diversity of microorganisms in soil, the microbiota populating the root soil interface is not randomly assembled. There are a series of checkpoints that I highlighted here in this slide that ultimately determine the microbes that populate this particular interface. Most of those microorganisms, I would say the majority of these microorganisms, are acquired horizontally, so from soil. Soil represents one of the main sources of inoculum. And within soil, the activity of farmers or the interaction among microorganisms, again, start to determine microbes that can interact with plants. But it's undisputed that an element of selection is the plant itself. And this observation underpins what is the central questions of our lab. Because if you get to know a bit better the molecular mechanism linking a plant to its microbiota, then we'll be in the position to rewire them to the benefit of the plant and, by extension, the benefit of mankind. Now, this is a biological process. Like any biological process, should be seen through the lenses of evolution. When it comes to crop plants, a hallmark of evolution are certainly the processes of domestication and crop diversification that progressively have differentiated modern cultivated varieties from wild ancestors. Now, there is evidence that within this framework, the activities that were essentially tailored to the modification of the plant and its genome, impacted on the capacity of plants of engaging with below-ground microorganisms. Now, because wild ancestors of modern cultivated varieties often thrive in marginal soil conditions, there is an area of particular interest in characterizing the so-called wild microbiota, because in there you may have the resources to basically cope with this marginal soil condition to the benefit of crops. However, I think that we need to keep an open mind in relation of this evolutionary trajectory of plant microbiota interaction, because even unintentionally, breeders may have already selected the optimum plant microbe combination for a given soil. And what we think is fascinating, the two scenarios are not mutually exclusive. For instance, depending on particular soil conditions, having a wild microbiota may confer an advantage to plants compared to, let's say, a more modern one. Now, regardless what is your flavor at this type of interaction, I hope that you agree with me that if you want to dissect the molecular basis of plant microbe interaction, perhaps we need to work with an experimental model, so a plant that is A, genetically tractable, and B, that has been subjected to these processes of domestication and diversification. And we believe that barley fits very well this bill for a number of reasons that you can read by yourself in this slide, not least the fact that wild relatives of modern cultivated variety are accessible for experimentation. And similar to many other traits that have been characterized so far, you can really investigate plant microbiota interaction within the framework of domestication and diversification. So we have defined the main actors here. One is the microbiota populating at the root soil interface, and the other one is barley. So let's have a look how we study this interaction. And in a very simple term, I think that we can identify four main pillars on which, let's say, plant microbiota studies can be conducted. The first one, like let's say any plant, let's say any plant trait or any plant characteristic, starts with growing the plant itself. In particular, for the study of the barley microbiota, we tend to concentrate our activity under controlled environmental conditions. So our idea is really focusing on the interaction between plant and soil and leaving out potential confounding factors represented by other environmental variables. We are particularly fond of so-called common garden experiments. So what we do, we have, let's say, a reference soil or a soil with different, let's say, characteristics that we want to test. And we use this as a substrate, basically in a glasshouse experiment where multiple genotypes, multiple conditions can be tested side by side under the same condition. One of the, let's say, and essentially what we do is that at a given developmental stage, we physically uproot the plants from the pot, we give it a good shake, and what we are left with is a thin layer of soil surrounding plant roots that can be, let's say, effectively removed only with some chemical or physical procedure. And for us, this is the operational definition of the rhizosphere. We have also, alongside our plants, what we call unplanted soil control, which is exactly what it is. So a pot that has been, let's say, placed side by side with plants and subjected to the very same condition and will be processed in the very same condition. So this is something that provides us a sort of baseline microbiota to assess what is the impact of the plant on those communities. And you'll see in the reminder of the presentation that often some of the figures report the let's say, the name bulk or bulk soil. So you see that this is the, let's say, the unplanted soil control. Once we have separated the fraction of interest containing the microorganisms, the microbiota of interest for us, we proceed with the characterization. And here there are two main ways in which the microbiota can be characterized. One, and basically the differentiation is whether or not you isolate microorganisms from that particular substrate, or you process with some, let's say, or you process or characterize microbes using some of their properties, very often their DNA. What I would like to stress here is that these two approaches, are let's say, is not a dichotomy. Sorry. So the approach should be not the one at a football rivalry, is not either or, but they complement each other, and both of them have their own pros and cons, and then you have some situation in intermediate. But regardless of the approach that you use for the characterization, the endpoint is almost invariably the same. A significant amount of data in form of multivariate data set. The majority of them is sequencing data, so sequencing of the microorganisms in a given environment that very often, or should be actually, often accompanied by what are defined as metadata, so the characteristic of the experiment, the characteristic of the plant. And to essentially distill biologically meaningful information out of this data set, let's say computational approaches are required, and this is the last pillar of our analysis. And for the, let's say, the person, the audience that are, let's say, particularly fascinated by this kind of science, I pasted here the address of the GitHub page of our lab. You can go there and visit and see physical description codes we have used to generate figures and stats. In the reminder, the reminder of the slides, very often here I will include also additional information regarding the interpretation of the data from, let's say, stats or analytical standpoint. Okay, now this is the way we study the microbiota, and the most popular way to study the microbiota these days is the characterization, especially if you're interested in bacteria, is the characterization of the so-called 16S ribosomal RNA gene. So let me spend a couple of words on this phylogenetic marker. Essentially, this gene is widespread and conserved among bacteria and archaea, and has a very interesting feature that were firstly described, or at least described accurately by this eminent microbiologist in the 70s that used this 16S ribosomal RNA gene to essentially demonstrate the existence of archaea and a reelaboration of the tree of life. So I would say the most striking feature of the 16S ribosomal RNA gene is that there are areas of the sequence that are conserved across species and would be the valley in this sort of roller coaster plot. And there are areas that are actually highly polymorphic and diagnostic for the identification of certain species, and will be the peaks. Those peaks are called hypervariable region. And basically back in the 70s, when Carl Wolf was studying the ribosomal RNA gene in microorganisms, he was confined on basically radio-labeled probes to characterize this molecule, and that would have been obviously not very high throughput. Already in the 80s, the development of the PCR, scientists made immediately the connection that by designing primers in the conserved region and amplifying the fragment, let's say, of an hypervariable region, the capacity of the diagnostic capacity, the capacity of, let's say, increase the throughputness of analysis increased. But the real boost for microbiome science were the introduction of next generation sequencing, particularly in the second half of the starting from the second half of the year 2010. This, coupled with the amplicon approach, became the I would say the workhorse of microbiome science, because it enabled scientists to investigate microbial communities in multiple environments at an unprecedented depth. So there is a methodological innovation, but also there is a very conceptual, let's say a very simple conceptual approach. And let me explain this. So this will be an hypothetical microbiota with three samples that may be three different Parley gene types. And the cartoons here depict different bacteria associated with them, color-coded according to their taxonomy. So these are hypothetical real microbiota. Thanks to the 16S ribosomal RNA gene amplification, we can dial into these bacteria without the need of cultivating them and obtain a reasonably realistic picture of this community using the sequencing reads assigned to the individual taxa as a proxy for community composition. There is no need really to familiarize with the concept of ASVs. If you are into microbiota science, you know what it is. If you are not, for the sake of this talk, is just these represent different species, and essentially the different, let's say, the different blocks here reflect the different abundance. And this is an important concept that will follow throughout the presentation. So this is a quantitative measure that can be computed and analyzed using a statistical approach like any other quantitative measure. Okay, so this is the conceptual background that we followed or allowed us to characterize the microbiota. Let's see some of the results we gathered with barley, starting with the characterization of the wild and domesticated microbiota. And this is a snapshot from a study of a couple of years ago where we were interested in investigating the so-called the B1, what is defined as a B1K collection. This is a collection of wild barley that is ecologically referenced in the Southern Levant. And our question was, what if we grew some of these wild barley alongside the leaf variety, again in a common garden experiment? Will they recruit the same microbiota, or there is any, let's say, any suggestion that the evolutionary pressure they left as had a footprint in their mind? This is one of the results that we have gathered. So we are looking here at a representation of the microbiota. And let me guide you through, let's say, what this plot represents prior to discussing the implication. So what you are seeing here are the individual samples that we have sequenced and belonging to either the unplanted soil, and this will be here, or a genotype of these, let's say, wild barley ecotypes or elite variety, color coding according to the scheme that I have here in the right-hand side of the slide. Now, here we are looking at an individual sample, and the position of this shape in the plots reflect the degree of conservation of their microbiota, which implies that if two shapes are close to each other, they have a lot in common in terms of microbiota composition. While if two shapes are more separated in the plot, they have less in common, either different microbes or same microbes in different amount. Now, with this key of interpretation in mind, I hope that you agree with me that what we could see is what microbiota scientists call rhizosphere effect, which is a sort of structural diversification of the microbiota associated with the plants, the rhizosphere of the plants, and what is the unplanted soil. And for us, this is a hallmark. So we see this in basically any study, and this reflects what I show you in one of the introductory slides. So the capacity that only certain microbes can proliferate at the soil interface. Now, if we focus on the right-hand side of the plot, what we can see is a separation in color, and in particular in market for the group that represent the wild desert barley and the group that represent the elite one. And then we can apply a suite of statistical tests and ultimately conclude that on the basis of the composition of the microbiota, we can separate wild desert barley from the elite one. So there is a sort of diagnostic effect. But the important point also is that it's not just a wild and an elite. Within those groups, you may have more or less similarities of the microbiota, and therefore this may have implication for contribution of the microbiota to growth, development, and health. So this is a way in which we can, for instance, investigate the impact of domestication on the barley microbiota. Another way is asking explicit question. So within these processes of domestication and crop diversification, and in particular the selection of elite varieties since the 60s of the 20th century, so plants have been always selected to respond to high input in the environment, particular nitrogen application in the environment. Conversely, wild barley have not really experienced this type of stuff. We asked if we supplement plants with different level of nitrogen, what will be the impact on their microbiota? And essentially, when I refer here to N0%, basically means no nitrogen added to the system. So plants rely on what is present in the soil, while this N100% reflect basically the optimum dose calculated for elite variety of barley, and 25% is a quarter of it. This is important because in the remainder of the presentation, you see these shapes, this sort of shape code that will be maintained depending on the fertilization regime. Okay, so very briefly also on delving into this type of experiment with another representation of the microbiota. Again, please let me introduce what this graphical output represents, and then we'll discuss the implication. This is another representation of the microbiota, and here we are not looking at the individual samples, so the main plot, but rather at the number of bacteria differentially regulated, so differentially recruited between genotypes. The shapes reflect, as said, the nitrogen level and the color of the genotype. So we are looking, for instance, at the bottom part of this graph, these squares is a comparison between the microbiota of elite variety exposed to no nitrogen and the microbiota of a wild barley from the desert area exposed to nitrogen. So now, with this scheme of interpretation in mind, we can have a look at the impact of nitrogen application on the genetic control of the barley microbiota. And what is striking is that by looking at the essentially at the condition with no nitrogen, we do see a significant diversification of the microbiota, which is aligned to what I showed you before. The elite microbiota is distinct from the wild one, and in particular, the elite is more distinct from the desert one. However, we're talking about the same genotypes, the same glass salts, the same sequencing library, so basically uniformity procedure. When they are exposed to, let's say, the optimum nitrogen supply, let's say the impact on the diversity, if you want, of the microbiota is almost obliterated, suggesting that there the genetic control, there is an element of genetics that is hardwired in the genome of barley, but this is modulated by environmental conditions, so similar to other quantitative traits that you may have in plants. And we were particularly interested in what's going on at the level of low nitrogen, because as I mentioned before, this is mimicking what would be a marginal soil. So perhaps the wild microbiota in that condition is, let's say, thrives in wild barley in that condition thrives better, and you may have function in the microbiota that help plants in doing that. So then we decided to look at the functional characterization of the microbiota, which is called metagenomics. So from operational standpoint, this is distinct from the amplicon approach I referred to before, because there is no PCR, there are no selective PCR involved here. You try to sequence the totality of the DNA. What you obtain is a series of fragments, and what you want to do is try to piece them together to see what they are coding for, and ultimately try to, let's say, reconstruct the individual microbial genomes, and then try to infer what is their potential on the basis of whether or not are particularly abundant in a barley genotype or in another. So conceptually, these people that are familiar with genome sequencing in barley, this is probably what you. This is very similar to what also has been done with plants. The difference here is that we are trying to put together multiple genomes at a time, with the advantage that, unlike plants, the genome of a microorganism, particularly bacteria, is between 5 and 10 MB, so something more practical. But regardless of the conceptual scheme or the operational scheme or the jargon that is depicted in this slide, the concept, this type of investigation, is identical of the amplicon sequencing survey, because similar to what I showed you before, we can characterize individual microorganisms on the basis of their genome and their, let's say, their potential or metabolic capacity. And if you are able to sequence them and annotate them, then we can compare on the basis of that. And therefore, we have an in silico reconstruction of the microbiota based on a given functional annotation. Similar to what I showed before, the individual sequencing reads assigned to a given functional category becomes the element for quantification and characterization among samples. And similar to the amplicon sequencing survey, you may end up with plots like this one, where the distribution of the samples is now dictated by the abundance, presence, absence, or quantitative abundance of given function in their microbiota. So this graph is telling us, again, that we have a diversification between the unplanted soil and the rhizosphere. And then within the rhizosphere, we can clearly separate the elite one from the desert one on the basis of the function of their microbiota. And you can basically try to identify these functions. So these two graphs here, you are looking at two sides of the same coin. On the left-hand side are the actual samples, how they relate each other. On the right-hand side are the functions underpinning, retrieved by sequencing, underpinning this diversification. We can basically, let's say, if we concentrate on this part of this plot, we'll have what is called rhizosphere effect. So what is enriched in the rhizosphere and differentiate the plant microbiota from the unplanted soil, and within the plant microbiota, the categories of genotypes that we have investigated. And this, for instance, informed us that what is required for microbes to thrive in association with barley is a series of functions that are typical for polymicrobial communities. So the adaptation of the plant immune system of the plant, the adaptation of chemical compounds that may release by the plant, and for instance, the capacity of interacting with carbohydrates that may be released by exudation. So this is a confirmation, a way of something we suspected, but doesn't really tell us much about the capacity of either microbiota or sustaining plant growth. So we decided to embark on a different experiment that could be considered alpha way between the cultivation dependent and independent approaches that I showed you before. And this is what ecologists refer as plant soil feedback experiment. So the concept here is that if you grow a plant in a soil, at the end of the cultivation, that soil will be enriched by the microbiota that those plants or those plant genotypes like. Now, if you can remove the plant and you use the substrate for the growth of a subsequent generation of plants, and in that case, we focused on the elite one because we wanted to gauge what could be the contribution of the wild microbiota to the growth of a modern genotype, then we may be in the position to assess contribution of microbes to plant performance. And in that particular case, we have also introduced an additional control where we treated the soil to essentially disrupt the functionality of the microbiota or the functionality of the endogenous barley microbiota, and to see what extent may impact on plant feed performance. If we use above ground biomass as a proxy for plant growth, what we have seen is that indeed plants that are growing in heat-treated soil are not very happy, or let's say are definitely less happy than plants that are growing in a soil that has been treated as such and was used for previous cultivation of barley. Although we couldn't really see a marked differentiation between the soil conditioned by the wild barley and soil conditioned by the elite one. So this motivated us to dig deeper and try to see via sequencing what happened in this condition. This is what happened. Again, let me guide you through this graphical representation, and then we'll discuss the implications. So we have seen here the microbiota of the individual samples that we have subjected to this experiment, color-coded according to basically what they are, either unplanted or cultivated, and whether they were exposed to an elite conditioned or a desert conditioned soil. And what we were asking is, what makes plants growing in the native soil different from the unplanted one and different from the heat-treated one? Because very likely, or at least there, we have the candidate microbes that may sustain plant growth under suboptimal conditions. We identified a set of bacteria fulfilling those criteria, but what we were looking, let's say, when looking carefully at their taxonomic affiliation, what we realized is that regardless of the input microbiota, which had a distinct composition, taxonomic composition, at the end of cultivation, the microbiota of the elite plants seem to converge to the same point. So, in other words, plants act like an orchestra conductor, and regardless of the input, or at least regardless of the input in the condition we have tested, they try to recruit the very same microorganisms that we speculate are required for optimal growth under suboptimal conditions. Now, this is an interesting observation, and probably among them we have potential beneficial microbes we consider for inocular preparation, but this really doesn't really solve our question whether or not wild microbiota is maybe a source of beneficial function for sustainable agriculture. And we realized that we are missing a link here, because we don't know how plants shape those communities. And until we don't know how plants shape those communities from a genetic standpoint, again, we can't design experiments and hypotheses to be tested regarding the contribution of plant microbiota interaction to sustainable crop production. And with that one, I would like to interrupt my presentation and leave the floor to Carmen, that will guide you through this experiment, not before having told you that we are changing color coding here. From the remainder of the slides, whenever you see blue or shadows blue, this will be the elite or the elite microbiota. Whenever you see yellow, this is referred to the wild barley or the wild barley microbiota. I close here. Up to you, Carmen.
Thank you, Davide. Can you see my slides? Yes. So, yes, to answer the question, can we link host genetic diversity with microbial diversity? We follow this strategy to use a segregating population and the elite parent, here represented in blue, and a wild parent represented in yellow. And this segregating population that will have ideally most of the elite parent genome with introgressions of the wild parent. And then we will use as phenotype the bacterial composition of the 16S gene ribosomal RNA. So in barley, we have a lot of genetic resources, and one of these is this backcross multi-parent nested association mapping population consisting in 25 wild barley genotypes crossed with Barge, which will be our recurrent elite parent. So we select family 15, whose wild parent is HID 144, and this was crossed with the elite cultivar Barge. And then then it was backcross, and for a series of selfings, we will achieve the segregating population. And with this family, we assess the microbial composition and abundance as phenotyping mapping to identify genetic regions responsible for the plant host microbiota recruitment. So the first step for a genetic mapping, we need to make sure that the parental lines, they have a contrasting phenotype, in this case, the microbial abundance. And this is what is shown here in this ternary plot, in which is represented in the vertex represent different microhabitats. So an elite rhizosphere, the wild rhizosphere, and the unplanted soil. Every point is representing wild microbial ASB, or the lower taxonomic rank that we can see in these metabarcoding surveys, and the size of the dot is proportional to the abundance of that bacteria. So in this plot is highlighted in yellow taxa that was significantly enriched in the wild barley, and in blue taxa that is significantly enriched in the elite barley. And we can see that this taxa is different. So the parental lines, they have contrasting microbial profiles, and in principle we could use the segregating population of these two for genetic mapping. So then, as I said, we use bacterial abundance as a quantitative trait for scanning the different barley chromosomes for significant associations. And this is the example of this family called Olophagaceae, which is significantly associated with one region in the chromosome 3H of barley. If we do this individually for all the bacteria that were differentially enriched between the parental lines, we can come up with this barley, the genetic map of barley determinants of the rhizosphere bacterial microbiota. So let me guide you through these circle plots in which I'm representing here the 7 chromosomes of barley. The gray connectors indicate the genetic map used for this genetic mapping. And then we have plot the abundance of bacteria at different taxonomic levels. So ASB, genus, and family. Then highlighted in yellow is bacteria that is enriched in the wild parent, and in blue, bacteria that was enriched in the elite parent. And adding another layer of information is the taxonomy of this bacteria, which is represented by different shapes. So we can see that limited loci, they support the microbial recruitment in the rhizosphere of barley under our conditions. And but we see here a region in the chromosome 3H, which is associated with abundance of up to 5 diverse taxonomically bacteria. So, yes, so this locus, we select this locus as a priority for validation, and we call it QTL rhizosphere microbiota composition in the short arm of the chromosome 3H of barley, and I'm going to call it locus 3H for simplicity throughout the presentation, representing 5% of the microbial reach. And as I said, this will be our priority for validation. So what we did for validating this locus, so we went back to the segregating population, and we tried to find a line that was heterozygous at the locus of interest, and then we self this line, and we obtain lines that have contrasting wild and elite haplotype at the region of the locus of interest, and then we also genotype these lines. So our line of reference will be also always the elite parent Barke, and then here represented in dark blue. Then we have represented in pale blue the line 12417, which is elite-like at the locus 3H and is 95% identical to the elite parent. Then our other line for comparison that has a contrasting haplotype at the locus 3H is represented in yellow, is called line 12452, is white-like at the 3H locus and is 93% similar to the parental line Barke. So I'm going to use this color code throughout the next slides. And I have to say that despite these lines are really similar to the parental line Barke, they are not near isogenic lines. So we have other introgressions in other regions of the genome. So once we have the genetic characterization of these lines, we wanted to really make sure that these lines, they have a microbial phenotype. And this is what I'm showing here in this canonical analysis principal coordinates, in which every point is representing one microbial profile in the rhizosphere. And the different colors, as I said before, they represent our elite and our what we call sibling lines, the lines that they have that are sister lines and they have contrasting haplotypes while in elite at the 3H locus. And in this type of graph, as David explained, as closer are the points, more similar are these profiles, microbial profiles, each other. So what happened when we have wild alleles at the 3H locus is that we have a shift in the composition that is significant on the microbiota composition. So we have a diversification of the microbial composition, and this validates our findings in the QTL mapping. So then after making sure that the lines having wild alleles at the 3H locus, they have a differential microbial composition, we wanted to see what type of genes are in the locus. So we wanted to select candidate genes, and then for this we use RNA-seq to identify these candidate genes. And again, we use our sibling lines and our parental line of reference, Barke. And for this, we use root tissue, and we harvest the 6 centimeter uppermost part of the root system, so this part here, which is the same that we harvest or we survey for the microbiota. So running this RNA-seq, we found that several genes were differentially expressed, and we can see that most of the differentiation, the different expression comes from the comparison Barke and the line with wild alleles at the 3H locus. And then we have also the regulation between the sister lines or the sibling lines. But we were interested in the intersection of these two comparisons, so in these 34 genes as a potential candidates. But for identifying candidate genes at the 3H locus, we have a series of criteria. So these genes must be genes expressed in root and mapping at the 3H locus, that they show a marked differential expression among lines harboring contrasting alleles, and or displaying high impact variance among genotypes. So with that, we could select 3 genes as a candidate genes for this microbiota composition phenotype. So the first one is an NLR, or a resistant gene, which is putatively involved in the recognition of organisms. And this was downregulated in genotype harboring a wild-like locus. Then we have an uncharacterized gene, which was upregulated in genotype harboring a wild-like locus, and also has a frame shift variant and a missing stop codon. And then the last, our third candidate, is xyloglucan endotransglucosylase hydrolase, which was not differentially expressed in the RNA-seq, but we could see by variant calling that has a frame shift variant at the prime end of the gene. So this is a schematic representation of the locus, and then these are the physical distance between the different genes and the location within the locus. So perhaps the more interesting observation is also that if we compare our candidate genes with what was retrieved from metagenomic surveys in the rhizosphere, so functions of the microbiota thriving at the rhizosphere, we can perfectly match this with our candidate genes. So the NLR genes or resistant genes match with the term pathogenesis, and the xyloglucan match with a carbohydrate metabolic process. So this makes us to be confident on the selections of these candidate genes. So as a conclusions of the overall presentation, we have shown that while in domestication barley genotypes host contrasting microbiotas, nitrogen availability modulates the host genetic control of the microbiota, and we have located and validated a major determinant of the microbiota composition on barley chromosome 3H. And with that, I would like to wrap up, showing you briefly a recent review that we have published in the lab, in which we propose different strategies for the deployment of the microbiota in breeding and selection. So we can use first hybridization and selection, where we have one gene involved in the microbiota with a large effect, and this could be the case of the 3H locus. Then when we have polygenic traits with small effects throughout the genome, we could use genomic prediction for selecting this germplasm that has interesting microbial traits. We could use also the redomestication. So this means that we can find some plants with an interesting microbial traits, and then we can modify key domestication genes so that they have the more interesting traits in terms of agronomy. And then we can finally use the candidate genes approach. So we will select 4 genes that are involved in microbiota. For instance, it could be resistant genes or biosynthetic clusters. And then also for finalizing in the lab, we have isolated members of the microbiota. So this is what David told us the other strategy. So this is a cultivation independent approach, but this is very important because together with plant genetics, we can infer causal relationships. And we have been doing this with bacteria in the rhizosphere in this collection here. And it has been the source of many interesting achievements for the lab. And then more recently, we have also we were interested in the fungal side, in the fungal kingdom, also part of this microbiota living in the roots and the rhizosphere of barley. And we have created as well a collection of this of the fungal members of the microbiota. So if we implement these microbial traits in the breeding and selection programs, we can anticipate impact in agriculture decarbonization, sustainable agriculture, and overall in food security. And with that, I would like to thank to all the members of the Bulgarelli Lab and also the ongoing collaborations in the team and the funding as well. And thank you for your attention. And we will be very happy to take questions.
There are a couple of questions in the chat if you want to go there and pick them up.
I will stop sharing. Yeah.
I take the first one. If I'm not, let me see, let me scroll. Yes. So the first question I can read in the chat is, thank you, David, for the presentation. So you're welcome. A question in relation, the question is from Slim Arfaoui. I hope I pronounced it correctly. A question in relation with the effect of nitrogen, which modulates the genetic control of the microbiota. And as shown in the graphic, that with low nitrogen, let's say in the system, there is high level of interaction. In this case, we advise farmers to do rotation by introducing legumes such as bean, peanuts, chickpea, which they give some nitrogen in the soil. How, let's say, how we proceed in this case? My answer would be, let's say, I think that the reason why we have done this experiment is more to understand what is the capacity of plants of, let's say, engaging or shaping their microbiota depending on nitrogen level. And probably the more immediate impact, the most immediate impact is not for farmers. So plants have to grow with some nitrogen, no matter whether with low nitrogen you engage more with the microbiota. But maybe for people interested in selecting varieties for low input agriculture, because if you select plants under, let's say, ordinary or high input, then you may miss out certain genotypes that can interact with potential beneficial microorganisms. But once this is, let's say, acquired in the field for the farmers, the advice is that provide plants with adequate source of nitrogen, whether this is organic and mineral, due to rotation with other legumes. So this is something that, let's say, is regardless of whether or not there is a specific interaction with the microbiota. The second question is there some studies in relation with allelopathy caused by barley? Not directly. So we have investigated a secondary metabolite called gramine barley, which is implicated also in allelopathic reaction. And basically, we studied what would be the impact of adding this compound in the rhizosphere and what would be the modulation of the microbiota, but only from microbial side. So we don't have, let's say, a direct link with potential contribution to allelopathy associated to soil microbes. Okay, so the next question is from Tim and Tim George at the IBH, and I lost it now. Yes, very elegant work. So thanks, Tim. What do you plan to do to further validate your candidate genes? I happily leave Carmen addressing this question.
Hello, thank you, Tim, for the question. So what we are doing now is really to decouple these three genes. So we have two strategies for doing that, and so the strategy is generating novel germ plants, and one is fine mapping. So we are trying to we back cross again our line harboring a wild allele at the 3H locus with the recurrent parental line to try to narrow down these candidate genes and break the locus. And on the other hand, we have a gene editing strategy. So we will try to knock out these genes and see what is the effect on the microbiota. And then we will do exactly the same surveys, or maybe yes. So we can use this in the same context or different contexts to see what is the effect of the locus under the same or different environments. I think you are muted, Davide.
Before taking the next one from the chat, there is an hand raised among the audience. I'm not sure if anyone that does their hand raise can unmute themselves and ask the question.
Hi, Davide, can you hear me?
Yes.
Okay, I'm Anastasia from the Minority Foundation. I don't know if you can see me as well. Thank you to you and Carmen for a very interesting work. So when you say you make the experiment with soil, you take representative soil from the field and put it in the pots. Do you envision that this kind of soil disturbance can have an effect on the functionality of both microbiota and how the plants perform? And do you have an idea? Have you any plans in addressing it? If so.
Yeah. Yes. Thanks for your question. Yes. We are performing what is called a reductionistic approach, and it's true that collecting the soil from the field may allow us to focus on, for instance, the chemical composition, eventually the texture, but then we lose what are let's say the structure, the macrostructure, due to the fact that it's been plugged or not, or left bare. Yeah. So in our experiment, we are not focusing or addressing these elements. So the idea, and we recognize that they are still important. So the idea is that once we have a better grip on our candidates or lines that we consider relevant for microbiota recruitment, those then can be tested under agricultural relevant conditions, including the one that you mentioned.
Thank you.
So I answer first the second part of the question. Let's say other than let's say mineral availability, the answer is no. So, or at least not yet. This may well be part of future investigation. That is obviously very relevant. Regarding the first part of the question, there are say several phylogenetically unrelated members of the microbiota that are significantly associated with locus 3H. And the very interesting exception is that among those, there are no actinobacteria. So actinobacteria seem not to map at locus 3H, at least when we have the wide allele. And probably this is the most striking feature taxonomy-wise of our locus. Now, there is another question. Probably Carmen can take this one. So, Carmen, can you see the chat? The question about the follow-up on exudates?
Yes. So we do test root exudates and at least the primary metabolism. We take exudates for plants grown in sterile sun, barley plant, and using this the same genotypes, our parental line and our sibling lines for comparison. And we check carbon content, nitrogen content, and then we went to GCMS and to do a more in detail characterization of the primary metabolism, and we couldn't find any differences in the exudates of these lines. Also, the candidate genes are not directly related to that, but it could be related. But we couldn't find this, at least under the conditions that we had. So we didn't use soil or the agricultural soil in which the phenotype was observed. So this may have impact on the exudates, but doesn't mean that we could have when we can have an impact if we are able to test this under more soil relevant conditions. And then also we didn't account for the secondary metabolism in this survey. So in principle, root exudates under the conditions that we use, so it was a sterile sand, they didn't show any differential abundant compound that could be related among the lines tested. I don't know if you have something to add, David, or?
No, I don't. I'm okay with these answers. I was just checking if there are other. Yes, there is another. What software databases do you use to assign function of different bacterial taxa? Okay, so a number of software and database. Actually, I was looking up at the actual paper, so I can give you the right answer because this is something that we have done in collaboration with our colleagues at the Division of Computational Biology. So, for instance, looking at the where is it? Yes. Regarding nitrogen biogeochemical cycles, there is a specific database. Tell you what, as an answer, probably is better if, let's say, I refer you to the actual manuscript, because this is not just one shot. Depending on what we were analyzing, whether it's shotgun metagenomes or individual mags, we have used different software and different databases. Everything is, let's say, described in the manuscript. I will send you the link in the chat, and there you can really get the information, the most relevant information. Metagenomics, as I said, is something that we do in collaboration with computational biology, and they are the real, let's say, the real mastermind of that. So let me pop the link so you have this information. But yeah, all I can see is that we tend to use the one that are, let's say, benchmarking for the field. So we haven't deviated from the one that you could find in especially the one for microbiota. We haven't predicted function from taxonomic information, if this is what you were alluding to in the question.
There are a couple of questions more.
Well, that's the next one, Carmen, is certainly for you. That was really interesting. Have you looked to see if there are any interaction between the fungal community and the bacterial community?
Yes, so we look the same graph that I have shown with the composition of the bacteria when we observe the phenotype with our lines with contrasting haplotypes at the locus of interest. So we survey the 16 spore bacteria and we survey the fungi in the rhizosphere as well, but we couldn't see a significant genotype effect, let's say, in the fungal community. So we thought that this is a bacterial phenotype, at least in the rhizosphere, what we can see in the rhizosphere. Maybe we need to look at other microhabitats and see.
Okay, so and a follow up on this question. So this is unrelated to the locus 3H, but we had a collaboration with the group of Alga Zucchero at the University of Cologne in Germany. But basically, we provide them with our bacterial collection, and then they probe it. Let's say the plant protection exerted by this community against Bipolaris sorokiniana, and they have identified such a characteristic in bacteria that we have isolated from the rhizosphere. But yeah, we can't tell whether this is, or at least we can't tell at this stage whether this can be mediated by locus 3H. A related question: have you, from Chris Stephens? So great presentation, thank you. Have you tried to confirm the beneficial detrimental effect of the microbial species reached by the locus 3H, perhaps growing a second generation of plants in the same soil? We are trying to do that, not in this way though, but basically by exposing axonically grow plants to a bacterial collection that is mimicking the composition or the differential composition we have seen in our survey, and see what will be the impact on the plants. We have just preliminary observation in this direction, so we can't really comment much on that, but this will be the line of investigation that we would like to follow to address this question. I think that that's all. So I don't see additional question in the chat. If I missed some of them, please reveal yourself or shout in the microphone. Otherwise, Donna, I think that we can close here, and I would like really to thank you, the audience, for having let's say joined us and listened to us and provide us with very interesting questions and feedback.
Thank you.
Thanks, everyone. Thanks, David and Carmen.